Hi, it's Dr. Barr, and I love me some histology. We're going to talk about basement membrane. By the end of the session, I want you to be able to explain the composition and function of the basement membrane and detail its characteristics at the light and electron microscopy level. So the term basement membrane is used to visualize the structure under light and electron microscopy. On the image of the right, you can see this amorphous, dense layer underneath the epithelial basal layer, and they tend to be variable in thickness. Here's an example here of the ciliated pseudocolumnar epithelium with the underlying basement membrane that's typically seen in the tracheal region. We can have special stains such as the PAS or the silver stain that can help identify basement membrane that tend to be thinner and surrounding the epithelium. So in the image here, you have your renal-tubular epithelium, and these special stains help make the basement membrane pop. In this area here, you have your glomeruli with a very thinner basement membrane. The basement membrane is comprised of two components, the basal lamina, which is your extracellular matrix that has direct contact with the basal aspect of the epithelial cell, and then you also have your reticulamina, which supports the overlying basal lamina, as well as is a structure that's continuous with the underlying connective tissue. It's comprised of type 3 collagen fibers. Now here's just an electron microscopy to show you the relative location. You have your epithelial cell in one region there. This gray dense line that's designated right here is going to be your basal lamina. And then you have your reticular lamina underneath. And then on the other side, you have your fibroblast nuclei, right, which is going to be the one making your collagen. With the basal lamina, there are components that I want you to be mindful of. The major components are your type 4 collagen and your laminins. The laminin is basically comprised of 3 disulfide linked chains, and they are binding sites to integrin, type 4 collagen, and anactin. There's a third molecule that I want you to be mindful of, which is cellular fibronectin. It's produced by fibroblasts and has 2 cross-linking protein chains connected with the disulfide bonds, and it's a binding site for integrin, collagen, heparan sulfate, and fibrin. Now there's a mnemonic that I want you to be mindful of, which is called laminated collar around the neck. The phrase laminated collar around the neck indicates laminin, collagen, fibronectin. Recall again, Dr. Romana had talked about basal lamina in cycle 4 of Foundations of Medicine. Just to remind you again, the functions of the basal lamina is for cellular attachment, compartmentalization, separating epithelial cell from other locations, a site for filtration or collection of soluble proteins, and we see that with the glomerulus. And the basal lamina also play a role in cell signaling and regulation, where you can see it in migration of neurons, tissue regeneration, and embryogenesis. With the basement membrane, aka basal lamina, another example is what you would see in the glomerulus. The function here is for cellular attachment, compartmentalization, and filtration. So here is a lower magnification where you want to look at this gray line here, which is going to be your basement membrane. On one end, you have your podocytes. Here's your basement membrane, and on the opposite end, you have your endothelial cell and your capillary lumen. At higher power, you have your epithelial cells on either side, podocyte foot processes, basement membrane, endothelial cell. Notice that there's no collagen fibrils or no lamina reticularis. The dark line in the center is called the lamina densa, which is comprised of type 4 collagen and fibronectin, whereas the lamina lucida, which are the lighter staining areas on either side, is comprised of lamina. Keep in mind that the lamina lucida is suggested to be a fixation artifact, and that's why it's lighter in color, but do know the compositions are very distinct between the lamina densa and the lamina lucida. Another example is the epidermis, where you can have your basal cells forming attachment onto the basement membrane, compartmentalization, where you have epidermis on one end and connective tissue on the opposite end. And when you're talking about a wound healing, like if you have a paper cut, right, the basement membrane serves a role as cellular regulation and regeneration. Now here you can see the keratinocyte is on one side. It's anchored to the basal lamina here, but via the hemidesmosomes, which are these dark linear structures over here. And these are just to show you the hemidesmosomes. Now, the lamina lucida is this light-colored linear area right here, and then underneath you have your lamina densa, which is your darker staining region right there. Now, keep in mind that you have underneath the lamina densa is what we call the sublamina densa, which is basically your underlying papillary dermis, aka connective tissue. So you can see that the cell is anchored down to the basement membrane via hemidesmosomes, and you can distinctly see the lamina lucida and the lamina densa. Don't forget what is the composition of the lamina lucida, right? And what comprises the lamina densa. Don't forget the phrase laminated collar around the neck. Basement membrane is a term used to identify structures on light and electron microscopy. It's comprised of the basal lamina and the reticular lamina. We use the basal lamina term particularly for electron microscopy. It is the layer that interfaces between the epithelium and underlying connective tissue. It's comprised of your type 4 collagen, laminin, and fibronectin, and don't forget about the key functions that it serves. On that note, please email me or contact me if you have any questions or need clarifications, and I will see you at the next lecture. Thank you. Bye!